for microbial infections, they get antiviral. Sometimes, I mean, I've even seen doctors or pharmacists giving away everything. So they would give you antifungal, antiviral, antimicrobial, because they're not sure which agent caused the disease, right? That would need proper investigation. And so what they do is, by giving you those medicines, they make your immune system weak, in a sense that if such a bacteria or virus was lying in your stomach and you're already defending yourself against it, that drug kind of makes you weak. Your body becomes weak. And if there was such a form existing, it would already develop that ability to fight the drug, and so become more powerful. Exactly like how COVID can at times learn to fight with a drug, learn to fight with the environmental conditions, adapt, because they're kind of dying every day and growing every day, or living every day. Especially for bacteria, they have such a shorter life cycle. And virus is even more difficult, in a sense that, like Daniel said, it's between living, it's at a borderline between living and non-living. It's not really a cell, but it's not really non-living either, because it contains proteins and DNAs and RNAs. So when it's in a living body, it's living, because it preys on its host and it replicates and multiplies and divides and forms more individuals, or increases its population. But when it is outside, it's just dead, right? So how do you kind of understand these things to make your day-to-day life more I don't know if I should say interesting, but interesting. Okay, so let's get to the presentation. And I'm way behind. All right, so all of you can see the presentation? Yeah, great. So today we will be looking at the, okay, participants are being added. I just want to make sure they are and that Summer is here. So today we will be looking at microorganisms and trying to understand their role in the environment. And unfortunately, due to the current times, most of you probably are already informed about what's going on. And I'm saying unfortunately because a part of it is also a blessing in disguise, where people are educated to otherwise things that would ignore. So flu viruses are not uncommon. We are used to people just walking into the office when they have flu. No matter how virulent the form, they would just come into the office and sit and share the space and cough around. So that sort of basic courtesy did not exist in public spaces in general. But I think now people are careful when they have even normal influenza, flu, or anything else. They make sure that they cover their faces or inform another person about it. And the surface microbes or surface viruses and microbes, germs that were lying all around us, is also not something new. It has been there. This area has been there. We've been seeing advertisements on, XYZ kills 99.9% germs and so on. It was always there. I think our eyes have opened because of something virulent taking over the entire planet, because of how the planet has changed. How virulent this virus is can be compared to how virulent influenza was when it first came, and how its reach increased because of then traveling and interaction and businesses. But of course, the scale is now much bigger, and the impact, therefore, is much bigger, and it has changed our attitude towards how we should react when such a germ or bug is around us. So that part of microbiology actually is called epidemiology, where you study diseases. In environmental sciences, we generally encouraged to look at the positive sides of positive side of the story. So we almost always told about the good things or the services that these microorganisms provide to us. So I'm trying to, because you all already giving some midterms, your midterm is close. I've tried to revise some slides, brought them back in this lecture, so that you would know why they were taught then, and then also you would know that how these things cannot be compartmentalized, how they cannot belong to a certain lecture, and that they kind of it would make sense about why I taught you a certain something in the previous lectures and how we could make use of that information here, even though traditionally it is not taught like this. So this was basically when we looked at the ecosystems, when we looked at the major components of an ecosystems, when we looked at a biosphere, past the cell. I told you that many cells combine together to form tissues and organs and therefore organisms, but there is a life form that's very primitive, but is oldest and has been existing on the planet for a long time. And they actually have their own mechanism. So they are unicellular or multicellular, but very small. So small that they're not visible with the naked eye, and that's why we call them microorganisms. And they have their own population, their own communities, their own ecosystems, except that they're not visible to us as these bigger life forms on the biosphere are. So now we know why we call them microorganisms, because they're not visible to the naked eye, and that's why we need microscope or another form of intervention. Even there has been so much development, because you have TEM or SEM, that's the scanning electron microscopy, that gives you very, very high magnification or very, very high resolution for something as small as a bacteria or a virus. And again, another slide here just for you to look deep into what actually a cell contains. It has to have a nucleus. For example, if you study about virus or COVID in general, the viruses do not have a nucleus. And that's why their placement is slightly hard. So they are put in the microorganism category, but they're not put in the prokaryotes or eukaryotes generally, because they don't have a nucleus. And that nucleus contains chromosomes, which contains the DNA, that contains the genes, and that genetic information has an important role to play in how we react to certain environments, as well as what we are and what we are represented to be. So our looks, our eyes, the color, everything, and that then relates to our activity on this planet. So these were both slides from the previous pictures that we had studied in the biosphere. But let's get back to what we are going to look at today, and why I thought it was important for you to look at it in the flow of series that we've been doing. Ideally, I should have taught this to you in the biosphere lecture, but I thought this would have been too much information for you there, especially for people who haven't studied science in a while. But now I think, because we've been mentioning things, and in future when we talk about pollution control strategies, microorganisms have a huge role to play there. That's why I had to teach it to you at some point. And if you kind of look at this chart, people have defined it differently. Generally, they just put bacteria, protozoans, fungi, viruses, and parasites into the microorganism category. All have certain roles to play. Some of them are unicellular, which means they have single cells. Some of them are multicellular, but most in one form or in one type are not visible to the naked eye. So bacteria, protozoans, etc. generally are not. But there are other parasites, such as or other microorganisms that are generally visible to the eye, but they've still been put in the microorganisms category, such as helminths. So helminths is visible to the naked eye, but its egg is not. And that's why it is being put in the microorganisms category. So if you want to study it, you kind of need a microscope to, or another magnification method to study it. And that's why it is being put there. When I put this picture here, and when I was putting it together, there is an extended biological definition and description to it, where I talk about the nucleus, I talk about the walls, but I thought I shouldn't put that information here now, because I think it would be too much information, and maybe I would need more biology for it. But students who have biology and chemistry background must have read this and should know exactly how one is different from the other. You have seen them. You've seen algae, you've seen fungi, algae and fungi, or you've read about bacteria, you've read about viruses. So you kind of have an idea of how these could be different forms of microorganisms. And their cell compositions could be different, their means of locomotion could be different, how they produce their food could be different. For example, algae is plant-like and because it can photosynthesize, fungi is not. Fungi cannot synthesize its own food, and that's why it's always relying on somebody else. It generally decomposes organic matter to get the food. And within these, it is said that there's still a huge area or huge number of species that haven't been discovered in there. And when we grow bacteria on the plate, it's said that only 1% of the bacteria present in the environment are plateable. And that's why advanced technologies are coming there as well. And now I bring back another slide. And this slide was shared with you in the hydrosphere lecture, where I told you how, depending on what the pore size of the membrane is, certain contaminants can be stopped. And here there was no mention of microorganisms because I hadn't taught microorganisms to you. So I just said that as you move from microfiltration to ultrafiltration to nanofiltration to reverse osmosis, you can get rid of bigger suspended particles, macromolecules, bigger molecules, smaller molecules, until you get to that finest stage, which is the nanoscale in reverse osmosis, where you only get clean water. Now, in this particular lecture, I think I asked you to look at the water filtration setups in your house or in your locality or in your university and find out where exactly does it lie. Did anyone do the home task, or do they remember exactly what kind of filtration was in their water filter in the home? Or did they ask anybody around? Hassan, have you? Okay. Why is it the usual suspects? Huh? Hassan and Rafi and... Okay. I don't want to waste time, so maybe let me go with Hassan then.
So I did look it up. I don't remember the entire procedure. I did write it down before, but I basically looked at the drinking water that we have. So we get it from Polygon. So I looked at their process. Like they have ultrafiltration and they use reverse osmosis to clear up everything. I had a detail, but I don't remember it right now.
Brilliant. No, I think that's enough information for what I was going to say. Rafi, do you want to add to this?
Yes, same. हमारे घर में भी वो reverse osmosis हो रहा था उस machine में.
Perfect. So you know that that filtration mechanism technically gets rid of all the ions, even monovalent ions, and should only provide you with, ideally, should only provide you with water molecules. And what does that mean for the microorganisms? Because, so like I said, there I did not explain it because I hadn't explained microorganisms to you. So if you ignore everything else, right now, these days, you perhaps want to know whether the water filtration you have in your house or in your community can get rid of bacteria and viruses, right? So in order to get rid of bacteria and viruses, you need to go as fine as for bacteria's microfiltration, and for viruses, ultrafiltration. So if you have up until ultrafiltration, ideally, whatever's in the source water should have been gotten rid of. But that doesn't mean that the filter water that you have cannot get bacteria from the environment. So if, let's say, in the piping system or somewhere else, there was another pathogenic bacteria that can still enter it, but whatever was in the source water has been gotten rid of. And so, you can kind of look. So for example, let's say that the nanofiltration membrane is in the range of 1.1 nanometer, and you can see that it gets rid of practically everything, from a frog egg, from a human cell, from a plant cell, down to lipids, for example. Rafi, do you have a question?
I just wanted to ask because like मेरा science थोड़ा weak है. Do you think जो कि calcium और जो भी ions होते हैं पानी में वो filter से आके तो फिर वो निकल जाएंगे जिस filter में reverse osmosis हो रहा है?
हाँ, तो आप पिछला जो lecture हमने देखा था ना, पिछली दफा जब आपने किया था, let's say calcium is a divalent ion, right? It is a Ca+2. And where have I placed the divalent ions? before nanofiltration, it means that past nanofiltration, there will not be any divalentines. So the water that you're getting from your community would be, if it's reverse osmosis, would lack calcium and magnesium. And that's why people generally drink water that's provided from the companies, because they past reverse osmosis, like I said in the lecture in hydrosphere, they would add all of these minerals and mineralize it. And sometimes they advertise it differently, saying we've put all the minerals that you would get in Swiss Alps or in the north, or as good as a water that could be coming out of a spring from the mountains. So that's the kind of understanding. But is this clear? Because I think this is important in your day-to-day lives, in your general lives, whether or not you are a chemist or a biologist. I think this information is important. So if no one has any question here, I can move on. But basically, we said there are different levels of filtration. The scale varies, and at each scale, a certain something can be gotten rid of. Now we can purely look at it from a chemical perspective, which we did in the lecture on hydrosphere, or we can look at it from a biological perspective, because we're worried about diseases these days. And at each type of filtration, we could get rid, because it gets finer and finer, right? So from microfiltration, where it would be in the micro range, you would get rid of the suspended matter, biological or otherwise, and finally get to RO. And RO stands for reverse osmosis. Okay, so micro, as the name indicates, it's just 10 raised to the power and so on. So RO is more or less in the nano scale range, but very low nano, and even past that. All right. And again, this is something that I kind of brought back into the discussion. We looked at this last time. We have, I think Fateh in the very earlier lectures mentioned how cows or farms where a lot of cows are being farmed could produce methane. You've all looked at the biogeochemical cycles in your first assignment. It was kind of to bring back the diversity and the diverse environments in which these microorganisms could thrive, and the kind of actions these microorganisms could perform. Now, in the rumen of or in the stomach of the cows, the degradation or the digestion process is slow, and that's why you expect the bacteria to digest it for the cows and then release methane gas in the process. So to provide those nutrients as well as to benefit from it, this function is performed by a group of bacteria called methanogens in the stomach of cows. And a lot of research is happening in this area as well. And in the last lecture, we looked at wax worms, and we looked at how Enterobacter or Bacillus species could grow in their stomach and degrade plastic. We also know in the current times that how bioaerosols or pathogens that are contained in the breath or in the droplets of a sick person, when they cough or sneeze, can be transferred to another individual that's standing close to them. So they have a diverse environment, they have diversity within them, they can live in many habitats, as extreme as a hot water spring. And they have both positive and negative roles to play. So they can be very beneficial for the host, or they can be very harmful for the host. And that brings me to this discussion. So in the biosphere, instead of giving you examples, I just want to introduce you to these 4 terms that are very recurrent in microbiology. And I think for your general life, they would give you a good English vocabulary as well. So symbiosis is the process where both partners benefit. And this is not just microorganisms, but all of us, wherever we are benefiting something and that's benefiting us in return, we can call that relationship a symbiotic relationship. And it's kind of one depending on the other for living. So without one, the other would die. So let's say if fungus was depending on organic matter, and if the organic matter was missing and it would die, that kind of relationship would be symbiotic. Mutualism is just benefiting. So for example, E. coli in our stomach or in the stomach of most animals provides us vitamin K. in exchange for the nutrients that we provide to them or the food that we eat. Commensalisms kind of, one benefits, the other gets unaffected. So if yogurt is produced, then lactobacillus benefits, but and something along those lines in the cow's stomach. And parasitism is where the current situation where one is benefiting. So a virus is benefiting by growing on our body and multiplying in large numbers, but we are suffering because it's pathogenic and it's causing a disease and it's making our life difficult. Hassan.
Yeah, ma'am, can we say that corona is a form of parasitism?
Yeah, corona is definitely, all pathogens are parasites, as I've written. So all pathogenic forms, all parasitic organisms would be, and COVID is a parasite, right? Because it doesn't survive outside of a human body. And the proof for it to be surviving in an animal hasn't been proven yet. So unlike MERS, which could survive in camels, or SARS, which could survive in pigs, for this, the host so far, I think, is only human. I mean, the research is still happening, but so far it's definitely only human. Even though it has been acquired from, so it's called zoonotic because we have acquired it from an animal. And I did not put this term here, so thank you for asking this question. So it's called zoonotic because we acquire it from an animal, but past that we spread it amongst ourselves, and there's no other host involved in it. And that's why a lot of people who are pro-environment negotiate on the ways in which we exploit our environment, especially for meat and food and our general protein intake. But yeah, it is a parasite. And we were talking about habitats. I wanted you to speak more in this lecture because you have done your biogeochemical cycles. So maybe in one of the slides, I can let most of you speak. But just to give you an introduction of the different roles that they have to play in our environment. And I've majorly focused on bacteria, fungi, and viruses to just discuss the beneficial and harmful impacts with reference to each. In general, bacteria and fungi have both a lot of positive role to play as well as a lot of negative roles to play in their respective surroundings. In soil, they are a source of provision for organic matter. So they take their part in degrading the organic matter that's present there. In the nitrogen cycle, you must have looked at the process of nitrogen fixation where nitrifying bacteria fix nitrogen from the atmosphere for the plant that cannot do it themselves. And similarly, in the fungi, they can kind of connect two trees. They can connect two plants. Not only that, they can also increase the root zone for one tree or one plant when they're interacting with them. So they help them get the nutrients from the soil by increasing that root zone as well as connecting with the root of another plant or tree. So they are very important, of course, in maintaining the organic matter in the soil because they feed on it, they turn it into an available form for the plants. But remember, in the biosphere, we looked at bioindicators. And bioindicators were organisms that could tell you whether environment is changing, especially because of human actions, and how it's impacting the life, and would sort of be a wake-up call for us. These microorganisms, whether fungi or bacteria, are very sensitive to local conditions. So any change in the carbon concentration in the soil, any change in moisture, any change in temperature would affect their growth. So they can kind of indicate a healthy soil or an unhealthy soil. So not only are they essential, a keystone, but also they are a bioindicator as well, because if something goes wrong, especially because of our actions, they would disappear. Or their population would dwindle or come down. So just for you to look at. And I kind of, whatever I wanted to talk to, this particular slide has put it all in one form with reference to organic matter, of how microorganisms make it available, what is their abundance, how are they related to the nutrient pool, and what is their relationship to the plant. And it's not a very complicated slide, so I would let you go home and look at it. And in the aquatic environments, again, when we're studying hydrosphere, we looked at a typical lake, and we looked at the zones that could be developed there. there are different microorganisms, and the difference depends on how much light is penetrating, what kind of food is available, and what functions they have to perform. So if they have to release nitrogen back into the atmosphere, Pseudomonas will dominate, right? If they have to capture carbon and there's a lot of phosphate in the water, a lot of algae will grow on the surface, all blue-green algae called cyanobacteria. They would grow on the surface. A lot of litter that goes down into the benthic zone, there a lot of methanogenic bacteria, the same bacteria that are present in the stomach of a cow, would be present here and kind of degrading whatever dead organic matter lies there and releasing methane back into the atmosphere, or carbon dioxide back into the atmosphere. So just for all of this to be related to whatever you've been studying before, and complementing information that we've been looking at, that these microorganisms are found everywhere. They have a certain function in our geosphere, they have a certain function in our, they are of course the biosphere. So how they rotate nutrients and how they keep the environment as it would be is an important part of how environments remain in a shape, or try to maintain themselves over different changing times. And because so far the story was positive, I thought I will share something about the negative impacts as well. We studied particulate matters. Another deadly function that particulate matters have is that they act as a carrier for bacteria or for viruses. And of course, you know it because of the droplets that are coming out and COVID. But in general, the dust particles, the dust that's coming from a waste site could be acting as an agent to transfer pathogenic bacteria and fungi as well. So I think it's not very common here, but in general, in places where the environment is quite damp, what they do is, for example, in the UK, there's a lot of fungal growth. And that's why aeration and antifungal cleaning agents are abundant, because yeasts and their spores can be abundant in the air around you. And you kind of need to protect yourselves from them, especially if they're disease causing, or especially if the role is of food spoil, or if they cause spoilage of the food, then you kind of protect yourself from it as well. And it's not that, you know, these things have just evolved. This information lies there, it has been there, it has been studied. So for example, how much would you sneeze, how many droplets would you sneeze, what rate do you sneeze at, could kind of tell how far the infection would go. The size of a droplet could tell you how quickly it would disappear or evaporate, or go down and settle on a surface, or just die. So all of these, I think, are information worth knowing, regardless of this course and regardless of what we learn overall, because they would ensure your day-to-day hygiene and save you from the infections in general. And you always see in the advertisements that any antimicrobial or any antibacterial agent cleans or washes away 99.9% of the germs. What is the science behind it? I don't understand. Would any one of you like to talk about it? And it's can purely be a guess. And it's Rafe again. Go ahead, Rafe.
Ma'am, I think most of the germs are like washed away when when you're using any kind of soap जो आप lather बनाते हैं तो वो fats को dissolve कर लेते हैं और I think उसमें like most viruses तो उसी में मिट जाते हैं. So like they don't really need to add anything particular to take away 99% of the germs. Does that make sense?
Yeah. Aman, why can't you speak? Why can't you say what you've written with a loud voice? Is it difficult for you to talk? Should I just read it?
Okay, so ma'am, basically, I guess we can't kill like something 100%. So there's always uncertainty. हम हम श्योर नहीं कर सकते कि 100% किलो है, तो आई गेस जस्ट फॉर अनसर्टेनिटी के लिए उन्होंने 0.1 रखा होता है, बिकॉज देयर मस्ट बी सम जर्म्स प्रेजेंट इन द सोप जो किल नहीं हो सकते. सो आई गेस दैट्स द गेस.
Bilkul. And I agree, it's a marketing strategy. It's again one of those terms, you know. I think after this you should be very, very careful about what you read. First of all, when they say 99.9% germs, they already tell you that, you know, not all germs can be killed. And of course, how far have they tested it, right? And then I think one thing that they have to write it like this, because there are a lot of good bacteria, as we discussed in the beginning, and Hamza mentioned before we started the presentation, that there are good bacteria all around us and they're good for us. So we eat with our hands because we said the bacteria that are on the tip of our fingers help us in the digestion process, right? So if someone says that we're going to kill 100% of the bacteria, then another person could ask, but why would you kill the good bacteria? And so they kind of keep it lame by saying they kill 99.9% of the germs, so that everything's open to negotiation and discussion, just get away. And also, microorganisms are very, very, very intelligent. They can adapt to any environment, any pathogen, any drug that you develop for them. They can develop immunity over time, and they can develop the ability to fight that and grow regardless of what we can do. So that their ability to adapt, I think, encourages them to say that it's only 99.9% effective, because what if a bacteria or a virus has become more virulent and has adapted to what the soap can do. So yeah, and now again, this question is for you. All of you did your first assignment on biogeochemical cycles. You must have read somewhere about one kind of bacteria or another. I'm not saying you know the names, but let's say in the case of carbon, you said methanogenic bacteria, release methane back into the environment. Do you have similar examples for other kinds of bacteria that do provide certain services in the environment? Fateh?
Am I audible?
Yes, you are.
Yes. So in the nitrogen cycle, there's this thing called nitrogen fixing bacteria. They convert the nitrogen in atmosphere and any ammonia in the soil as a result of decomposition into nitrates. If there is a shortage of nitrates in the soil, and if there is an excess of nitrates in the soil, they convert the nitrates into nitrogen gas.
Very good, Fateh. You haven't studied science before, have you?
Just back in O levels.
Okay, I think I enjoy how much effort you put in learning the terms. So thank you. And yeah,
Thank you, ma'am.
you bring nitrogen from the atmosphere, you fix it, the bacteria fix it for the plant, and sometimes in the wastewater treatment process, they can use the ammonium there, make use of the nutrients and the energy by breaking it down and finally releasing the nitrogen back into the atmosphere. So Hassan or Hamza, somebody raise their hands.
Yes, ma'am. They can also play a role of bioindicators.
They can also play
I read an article in which they mentioned they are very easy to sample, and like if there's something wrong in the environment, they produce stress proteins inside them, and we can sample them and we can like look for it. If the percentage is higher, then there's something wrong with the ecology around them.
Exactly. Yes. Very good. Adeen. More hands.
Yes, ma'am.
Yeah, Adeen.
Yes, ma'am. In the assignment, I read that there are some bacteria which are very essential for producing oxygen as well. And if I remember correctly, the name was Bacillus and Lactobacillus, and like they contribute a lot in the oxygen of the environment.
Super. Thank you, guys. Anybody else? Okay, so I'll just jump to giving you two examples that I think would be relevant in your day-to-day life. So how many of you have gone to these mountainous areas or generally traveled around a lot of stones and seen very nice color patches ranging from yellow to red on the stones? Raise your hands. At least for this, you can raise your hands if you've seen this. A lot of you have, right? Do you know what it is? It's called lichens, and it's basically a combination of algae and fungi. And what happens is we know that algae can produce their food. They're plant-like organisms. They're not plants, but they're plant-like, and they can synthesize their own food. And fungi, on the other hand, has other abilities, like it can anchor to a site. It can absorb a lot of things from the environment because it doesn't produce its own food. So this particular combination is for them to kind of remain on those extreme environments and mutually benefit from each other. And not just that, they, I think, also render the environment really beautiful. I enjoy seeing the different kinds or shades of green, yellow, red on different kind of rocks and stones. And yeah, so that's one relationship, right? The other is mycorrhizal relationship, which generally contributed by mycorrhizal fungi. And what they do is, like I said, they can connect to plants. So they're relying on the plants for food, right? Because they're decomposers, recyclers, but not food producers. So they're kind of connecting to their roots and extracting some nutrients for themselves, but also creating that connection for the plants. And then, you know, depending on which type of fungi it is, they can perform different kind of functions in the environment. And so this association where they're benefiting from each other. And at times, as it says, it benefits because this is a beneficial species. It would stop any other disease causing fungi to come and attack it because it has sort of occupied the area. And I think this is a wonderful example, and perhaps would let you remember the kind of relationships that microorganisms or organisms in general have between each other in the environment, because these two are not micro. And I haven't put a lot about bacteria, even though I want to, but I did put an example of the kind of bacteria that could be found in wastewater. And I haven't put their names because I know it would be too much for you. But because you have read these terms in your biogeochemical cycles, I've put them here again. So as Fateh said, that nitrifying bacteria convert ammonium into nitrate and nitrite, and denitrifying bacteria release nitrogen back into the atmosphere. Some of them require oxygen or aerobic conditions, and some of them perform in the absence of oxygen or anaerobic conditions. And so methanogenic bacteria that are growing in the benthic zone or in the stomach of a cow or in a wastewater do not require oxygen to perform their functions. Polyphosphate accumulating bacteria, Nadia, I think last time asked how could we remove phosphate from the lake or pond, which is inside lungs. You could use, if there was a biologist in the group, I will totally say that they can use phosphate accumulating bacteria to do a research on how much phosphate exists there and how much can be accumulated onto a bacteria and whether or not it would be sufficient for the removal of the bacteria. So purely from a removal and remediation point of view. And sulfate reducing bacteria, perhaps you read this term in the sulfur biogeochemical cycle, where it reduces the sulfur from the atmosphere and kind of helps in the sulfur cycle. So just for you to know these terms, and of course I avoided mentioning the name of the bacteria. And that brings us back to viruses, because yes, their role can be very, very positive, and I can continue to talk about it because that's what I've mostly studied, but their role can be equally negative. And this image that I've chosen is from a report that the link for it is already given on the slide, is the fact that they're acellular. So when we said they're somewhere between living and non-living, and they contain certain characteristics of the living and some characteristics of the non-living, they incorporate themselves into the host, benefit from it, are of different kinds. So for this particular slide, and this is before COVID, they've shown COVID, one member of a coronavirus family, and another of Ebola. And I think both of them have created a lot of nuisance for people in all around the globe and in Africa in general in the recent times. So just for you to look at this. But back at the positive stories, a lot of functions that they perform for us, especially in food industry, especially in drug production, and by drugs I mean pharmaceuticals, is I don't think we could have been able to survive without them, or at least our day-to-day diet would have been impacted a lot. Yeast is a form of fungi. Aspergillus is a form of fungi. Lactobacillus is a kind of bacteria. And I've shown you, or the slide shows you how those particular yeasts and bacteria can convert one form into an, or give you a certain something that helps you in getting the kind of food that you get. And all of this, by the way, comes from the glucose. So the sugar is converted into pyruvate, and that pyruvate is then converted into all of these. Yes, Adin.
Yes, ma'am. You just mentioned like the advantages or like some benefits of fungi and the bacteria, but are there actually any benefits we get from viruses?
Yeah, unfortunately, no. Viruses are generally the bad guys. I've never seen anyone speak very highly of viruses. So in general, in life, they cause a lot of diseases. But now in the genetic engineering sector or in the biotechnology sector, phages are used for, I think, drug production or something. So they're for antimicrobial studies. So I think there is some use in the study, in the biotech studies, but generally in our day-to-day environment, viruses are only known to be disease-causing or pathogenic or parasitic agents. Yeah. And so basically in the food, why does your flour or the bread raise? Because of the carbon dioxide gas that is produced inside by these yeasts called Saccharomyces. And so it raises, and then you get that nice fluffy bread that you get. And lactic acid, which is a part of most of the dairy products, is performed by lactobacilli or aspergillus. And I've kind of expanded on it, given you a little more detail, just because I think you should know about this. Cyanobacterium is a kind of algae which is called cyanobacteria because it's microscopic and it has characteristics more similar to bacteria. But on the other hand, it's photosynthetic and it can produce its own food. I think, again, like it's one of those fascinating areas which you, or those species which you read about a lot in the field of environmental sciences, because they produce their own food. They are algae, but they have characteristics that are closer to a bacteria and are therefore called cyanobacterium. And in a lot of papers, you would read blue-green algae as their description. And they are used in pharmaceuticals, in food processing, in making the fuel clean. So remember how we've been discussing that hydrocarbons or the petroleum industry is very dirty. It produces a lot of contaminants into the air. These bacteria, because they can degrade those hydrocarbons, can kind of, by degrading it and then recombining it, get rid of the extra pollutants that are present in that mixed oil or petrochemical. And similarly, the aspergillus produces citric acid, which can be used in many pharmaceutical and household activities. Like the vitamin C advertisements in the makeup and cosmetic industries, citric acid is an important part of it. And I've mentioned the lactobacilli again, or this time I've just written bacteria, because lactose, which is an ingredient of milk, in the presence of water and bacteria can be converted into lactic acid, which technically is your yogurt. Okay, I think I will, so quorum sensing and quenching, I just wanted to mention it, and you don't have to really know about it. It's probably for the 1 or 2 biological sciences students who are here, is the way in which microorganisms communicate. And they communicate, like you would think that they are not as intelligent and there's no form of communication between them. But I think the way they communicate with one another using certain chemicals and the way they sense it. So even for these pathogenic bacteria and viruses, and even for those that we benefit from, they have a way to communicate with each other, and it's generally called quorum sensing and quorum pinching, but it's not to scare the rest of you off. I just wanted to mention it so that if you ever see it, you're able to understand what the article is talking about. Some individuals, and maybe one question. The book that I've shared with you has made an entire list of individuals who are taking part in studies relating to hydrosphere, biosphere, or generally environment. Although this list wasn't in that book, neither is this topic, but I wanted to mention some of them because I kind of thank them, and I'm kind of impressed with how intelligent they were to study what they studied in the 1850s, 60s, 70s. So, Van Leeuwenhoek was the first one who studied bacteria or microscopic organisms under the microscope. Louis Pasteur did a lot of good things. Maybe I can ask you what pasteurization is, because this is something that's mentioned in your day-to-day life, isn't it? So what do you think is pasteurization? Killing germs in milk? Yes. 99.9% germs in milk. Because your milk still goes off, right? So fermentation, we've mentioned it in the biofuel lecture as well. So I think you're familiar with it already. Kind of mentioned it with the lactobacilli. So microorganisms or studies relating to microorganisms were really difficult because there was a lot of contamination, and Louis Pasteur had a huge role to play in reducing those contaminants. But what did Fleming do? And this is also a service to public health. Alexander Fleming. A new hand, guys. Adhan, have you raised your hand?
Ma'am, probably he discovered the enzyme lysosome, but I'm 100% sure.
Okay, something else, something very groundbreaking relating to disease. Fateh, go ahead. You get the call.
I don't think that Fleming deserves credit for penicillin because he was simply at the right place at the right time. It was accidental discovery.
Yeah, but I think to be at the right place at the right time is also a lot of effort, Fateh, especially when I don't know how many people are sitting behind their screens right now. I think I am very thankful to Alexander Fleming for being in the lab, for leaving something open, for working on one thing and then suddenly discovering that another thing that was present in the air could have that property. So yeah, maybe you guys should go home and look at what Fleming did, because I want to hand over the floor to Summer to share her work with you, which kind of relates to the study that we had on plastic. And she will introduce microplastics to you. She will tell you what she did in her BS in microbiology from the Department of Biology in LUMS, and will probably give you another lens to look at things. Okay, now I can invigilate. Now I can see who's sitting in the class and what are they doing. Maybe switch off my video for a while.
Okay. So basically my work involved investigating the interaction of polyethylene, which is a type of plastic, polyethylene microplastics with bacteria, the isolates which were taken from a place called Haryara train and Mahmood Bhuti waste dump site in Lahore. So basically, to start off, I want to give a quick recap of what microplastics are. Basically, microplastics are plastic particles which are equal to or less than 5 millimeters in size. There are two types, primary and secondary. Primary microplastics are those type of microplastics which are already 5 millimeters or lower in size when they are released into the environment. So examples could include examples could include plastic pellets, which are used in the industry. It could also include plastic pellets which are used in facial scrubs or shampoos or detergents that are used in homes. And secondary microplastics, on the other hand, are plastic particles which get reduced to 5 millimeters or lower in size as a result of breakdown in the environment. So examples of this include pieces of clothing which get reduced to the size of 5 millimeters or less, or large plastic products breaking down as a result of UV action. Um, so basically, what I wanted to do was to see how I could use microorganisms to degrade the plastic, specifically polyethylene that is in the environment. So there are various organisms that are in the environment that we could use for bioremediation. There's bacteria, there's protozoans, there's metazoa, algae, and fungi. And from these, I chose to work with bacteria because they play an important role in global ecosystem. They can grow in a wide variety of habitats and conditions, and they have a wide range of environmental and nutritive requirements. So bacteria in general, as Dr. Fauzi also talked about, are single-celled microorganisms. They are prokaryotes, which means that they lack nucleus and organized cell structure. They're very basic sort of living things. Some are pathogenic, they can cause disease, while mostly others are non-infectious, and they have a wide roles to play, such as they can decay the various things that are present in the environment. They can do fermentation, and they are very important in nutrient cycling, as you guys have already studied that. And they can also do nitrogen fixation. So bacteria also exist in various sizes and shapes. As you can see that they exist as circular, in circle shape, and they exist in rod shapes, and there's spiral shaped, and then there's like vibrio, the comma shaped, and these are the various forms that they can exist in. So moving on to why I chose polyethylene specifically to work with. Basically, polyethylene is used rampantly by everybody, so it is a major cause of pollution. Specifically, polyethylene is used on the coast of Karachi, and because as a result of it being used on the coast of Karachi and it is next to the Arabian Sea, it greatly and adversely impacts the biodiversity in the ocean. And thirdly, it has a higher tendency to absorb environmental contaminants compared to other types of plastics. So all of these factors were the reason why I chose to work with polyethylene specifically. Then the methodology that I used was that initially, I took two types of microplastics, as I mentioned, primary microplastics and secondary microplastics to begin with. So for primary microplastics, what I did was that I isolated primary microplastic beads from a facial scrub. As you can see, the clean and clear facial scrub. Please don't use this because it has microplastics in it. So I isolated these primary microplastics from the facial scrub by means of filtration, and then I took a polyethylene bag and then I cut it into very small pieces mechanically. Then what I did was that there's a technique called Fourier infrared transformation spectroscopy, a long name. What it is used to identify different substances by means of the specific position of the peaks in the graph. So the first graph shows the standard graph for polyethylene. And if we compare that standard graph with the graphs that I obtained for primary microplastics and secondary microplastics, I was able to see that both of these, all three graphs match in the sense that the position of the peak is the same. There are three peaks, and the position of all three peaks is the same across in all three graphs. So in that sense, I was able to verify that indeed the plastic that I was working with was polyethylene. Then I moved on to select the bacteria that I wanted to work with. So as I mentioned previously, that we chose to work with bacteria from Hadyara drain, as the biology department was already working with bioremediation over there. So I chose some two bacteria from Hadyara on the basis of their ability to absorb toxic heavy metals. Bacteria can also absorb heavy metals. So the bacteria that were able to absorb the maximum amount of heavy metals, I chose those bacteria. And we haven't been able to identify specifically the names of those bacteria. So for our convenience, we named them 1M1 and 2D2. Then we moved on to choose bacteria from a waste dump site called Mahmood Puti, located in Lahore. And we chose the bacteria over there on the basis of the fact that we identified pieces of plastic that were very old and they were decaying. So we chose those pieces of plastic, and then we isolated bacteria from there. Uh, and then we isolated bacteria from there on the basis of the fact that the bacteria, there's this thing called nutrient agar, right? So it is used to grow bacteria. So we use that nutrient agar to grow all the bacteria that were found in the plastic that we chose from this dump site. So the bacteria that were the most dominant and that grew rapidly, we chose those bacteria, and we named them MB1 and MB2. So after we were done with choosing bacteria, we decided to incubate the bacteria with plastics. And for this, we also chose a sort of medium that we could use in order to incubate them. So that nutrient had all the other nutrients apart from carbon, so that we know that the sole source of carbon for the bacteria is the plastic that we're providing with. So after incubating the plastics, the bacteria, and the nutrients, we decided to analyze them after every few days to see how the bacteria were interacting with the microplastics. So we did a lot, like a couple of techniques, and two of these techniques were using microscopes. So as you can see, in one microscope there are blue particles given. These are primary microplastics, and the other is the secondary microplastics. So you can see that there is a sort of brown substance that is holding or joining the primary microplastics. These are basically extrapolymeric substances which indicate that biofilm is being formed and bacteria are interacting with each other. So in the first picture, you can see that indeed the primary microplastics are interacting with each other, and they indicate the formation of the biofilm. And in the second picture, you can see that there are sort of purple all they're inhabiting the surface of the plastic, and these are also bacteria that were growing on secondary microplastics. So the greater the number of dots on these plastics, the more we knew that bacteria is growing over the plastics. And the second image is basically from scanning electron microscopy. It also shows how the bacteria is interacting with the plastics. It shows us more clearly, and we can have a better look at it, how it is interacting. So again, in the third picture, you can see that there's a plastic bead, and bacteria are strongly attached to that bead, which indicates the formation of a biofilm on the surface of the plastic. And the second picture that shows that on in secondary microplastics, how bacteria are basically clumped together and they're inhabiting the surface of the plastic as well. So through these images and through the entire process, we were able to see and understand that bacteria interacts with the plastic. It does so by means of the formation of a biofilm, and which to interact more and which were not able to interact as such. Moving on. Wait a sec.
Samar, if it's stuck, you can just talk about it.
So, Samar, could you just mention the scale of the SEM that you are
Yes,
using?
it's stuck.
Yeah, don't worry about it. Just talk.
Sorry, could you repeat your question?
So, Samar, I can't see the scale
Okay, so the
bar
main finding
of
were the
the SEM.
fact you can't see.
The scale bar of the SEM pictures that you are showing. What's this like scale bar? It's micro.
Of course, Azhan, I'm going to put this question back at you. What scale are the microorganisms? What scale could they be filtered at?
So it's again, the name suggests it should be micro, but the idea is how deeply we are like looking the images of the
I
SEM.
think it
It's
should be
like,
1 to 10 micrometer, because you can see the
yeah.
microorganisms. So it should be between 1 to 10 micrometers. Probably
Okay.
5 to 10, yeah.
Okay, so after whatever my work, the main purpose is to identify the exact bacteria that I was working with, and then continue basic types in the middle of conducting the study of the waste dump site study. So then I plan to continue the study and investigate how with the microplastics that I was working with. And then I also plan to observe microplastic samples in different environmental environmental samples that I could, for example, water bottles, or even toothpaste, or different environmental samples that I could basically observe. So this is an ongoing study, and I plan to continue it in the coming days. So that's it.
Thank you, Summer. Guys,
Is there any questions, please?
I think the floor is open for questions for the entire lecture, but if you have any specific questions for Summer or a general discussion that you want on plastics, microplastic, feel free to ask it now.
Summer, please can you repeat the ending, like your final findings? I didn't understand that.
Yes. So my final finding is that bacteria does interact with microplastics, but the way different bacteria do so is different. Like Hadiara drain Mahmoud booty side bacteria interact in similar ways, but I was able to observe that the Mahmoud booty side bacteria were able to interact more. And this could perhaps has to do with the fact I also did a test which is called test. Hydrophobicity is basically testing what material are more water loving and what bacteria are not water loving. So yeah, so basically this has to do with plastic, since plastic is hydrophobic, right? It does not dissolve. So bacteria that is hydrophobic will interact with plastics more. So I did a test to see which bacteria are hydrophobic and which are not. And according to that, the bacteria from Mahmoud booty waste site were more hydrophobic compared to the Hadiara drain bacteria, right? So after doing that test and after doing my study as well, I was able to observe and indeed confirm the fact that the Mahmoud booty bacteria were able to interact more with the plastics compared to those of Hadiara drain. So my study is basically, it was stopped because of the coronavirus. So I plan to continue my study in order to see how does it interact and at what rate it can actually interact, and if it eventually can degrade the plastics or not.
By interact, you mean decompose, है ना?
Yeah, first they will form a biofilm and then they will go on to degrade the plastic.
Okay. Thank you.
Yes, no problem. Any other questions?
Yeah, I had a question. How can we use these bacteria which you have identified at a larger scale?
At a larger scale. So basically, this is the preliminary step, right? After I understand how they interact at a micro scale, will I be able to understand how I can manipulate them and make them perhaps degrade plastic at a larger scale?
So, Samad, did you get a chance to observe any kind of rate of interaction so far?
No, I haven't been able to observe the rate. It was just understanding how they interact and uh, how different bacteria interact, like the process. And then I will eventually go on to find out the rate at which they can degrade, because degradation ke liye you need more time. The time was the limit in my study.
Okay.
Yes.
So, is there any specific sites like the specific functional group that are interacting, or like making the plastic degrade?
Specific functional group. As such, no, because since you're a chemistry major, I showed you the graph of FTIR, right? It basically shows कि different peaks कौन सी हैं उनकी position. If only मैंने FTIR भी बाद में किया था and the position was not changing, which means कि उसका functional group में change नहीं आ रहा था. So yeah, so it does interact with some sort of functional group, and by means of which I will be able to know if it's being degraded or not. So it's basically obviously polyethylene है तो उसी के साथ interact करेगा जो CH वाला part है.
लेकिन वो changes नहीं आ रहे तो that's mean कि कोई अभी तक effect नहीं है उसका. Because
हाँ वही तो
ideally
मैं कह रही हूँ, it's not being degraded as of yet, it's just interacting.
Samar, can
so
I
how
So
did
when
you
it,
conclude that they are interacting?
yes, Dr. Faudia.
Azhan, as Samar mentioned, this was a final year project, and you know, bacteria can take a lot of time to degrade, and we know that plastics take a lot of time to degrade,
Yeah,
and the
I get
bacteria
it.
that are degrading it take a lot of time to degrade as well. I'm not done yet. And so basically, when we looked at SEM, you know about these things better. It's just a surface imaging, okay? It's not telling us anything about the composition. It's only telling us the fact that the bacteria is adhering to the surface and perhaps is trying to do something, right? And that's why we never called this study a degradation study. We called it an interaction study because we just wanted to know whether bacteria actually go and attach themselves to the plastic. So in that simple time, we wanted to see what happened. And what the update that I can give you now is that I am speaking with the faculty from physics to see, because you know, and perhaps the physicists know, that this is a solid, plastic is a solid, but it is an amorphous solid, right? So it changes shape. So to see how the plastic was before and how it changed after the interaction, SEM or compound microscope wouldn't have been enough. So we're going to look at, using his microscope, we're going to look if there were any changes in even the way they were arranged. Forget breaking the carbon bonds, because of course we want them to feed on the carbon and therefore degrade the plastic, right? But forget the carbon bonds that we want to be degraded. We just want to see if there's any change in the way they are formed. So maybe that will then eventually expedite the process of degradation. So we're trying to study it over the time that it would take for degradation. We were in no hurry for it to be degraded. So we're just establishing basic knowledge, because such a study hadn't happened in the bio department before, or in the chemistry department for that matter. So I think we just wanted to start something new, but also understand the limitations of time and the limitations of space and gadgets available to us for analysis. But as we're learning about them, as we're borrowing more time, we are able to do more things. And hopefully at some point, we would turn this study into a degradation study and not just interaction study. But yeah, that is in the loop, and we would definitely do it. So as a supervisor, I have to come in and defend her. Okay, any comments?
So like we continue. So there's a difference in morphology or topological changes in the same image.
Azhan, your voice was breaking.
Okay, so I repeat my question. I was confused regarding the fact that they are interacting. Like how you guys they are interacting. Is it based on the same images, like the morphology or like topology changes, or like on what basis you concluded that they are still interacting? But like I'm just want to comment on this part.
Yeah, so basically, there is another fascinating area within microbiology, which is the biofilm growth, right? A lot of these, so when you study plastic stunts in your body or any other plastic equipment that you install for any deformation, etc., you want to see whether a contaminating bacteria would adhere to it and therefore cause a problem. All right. So biofilm is just a bacteria trying to grow on something. I think that's already step one of the interaction, right? The first step is that two different things come together, meet and interact. Okay. So that was the first step of interaction that we want to look at, that whether or not these bacteria are able to adhere to the surfaces of plastic. And then within biofilm studies, that's then again an extended process of studying where on top of one layer of bacteria, another layer is formed. Within that thin film, there are both aerobic and anaerobic bacteria that are able to grow. So the ones on the surface would be oxygen loving, and the ones inside those would be anaerobic, and they are generally more quick in degrading plastic. So if we leave that interaction study for longer, had we left it for a longer time, and had COVID not happened, we would have been able to see something more than that. But this study that was supposed to end in June, we had to end it in March. So the little that we could study was perhaps to establish the first step on whether or not they are growing, if they are growing, how they are arranging themselves, and how that arrangement could mean something. And we were able to find filaments of plastic on the compound microscope study that we did, just the simple light microscopy that you do. We were able to see filaments of plastic. Now, whether that was because of bacteria or simply because of the agitation, Samar and I weren't sure. But I think for those kind of things, not only do we study it for a longer amount of time, but also multiple times. So you, as an experimentalist, you know that I can't just conclude that those filaments or smaller pieces of plastic were because of bacteria. Because if I replicate it, and then if I'm 100% sure that that must have happened, then I conclude it. But yeah, I really think that we were still able to, by combining the chemistry and biology and environmental microbes, we were able to do something different that hadn't been done in the SSC before. So I still think that we were able to establish a basic interaction study, and their growth on plastic, I would still call it interaction, I think.
Thank you, ma'am. It does make sense.
Oh, super.
Best of luck.
Yeah, I hope that the study evolves, but with this, I basically, for example, you may not have thought about relating a plastic study with a bacterial study. So with this case study, I wanted, again, for you to think unconventionally beyond the traditional definitions of one thing or another, and beyond the traditional compatibility and degradation studies. Like, think beyond those boundaries, those compartments that we've created to explain things. Fateh?
Am I audible?
You are audible, yes.
Yes. So I have question regarding the economics